Hey everybody, it's Mr. Cuddill. We are almost done with chapter 5. Today's the last day. We're going to go over nucleic acids, and then I'm going to have you read a little bit on 5.6, which is using genomics to try to figure out how evolutionarily related two species are based on differences in their DNA and then hence their amino acid sequences. So anyway, we'll get there later, but right now, we're right here, 5.5. Some teachers don't choose to teach this right up front because we will be so heavy in DNA and RNA, all this nucleic acid stuff later in the course. Chapters 12 through 18 is all on genetics. However, I find it's good to get some of the basic stuff out of the way with students. And so I like to teach it. So enjoy. We will be having a test after this. It's just chapters 4 and 5 on this exam. One of the reasons why I like this unit is because it is applicable to everybody. You are what you eat. And basically all these lipids, nucleic acids, carbos, proteins, they all come into your body through your mouth. You get them into your digestive tract, you break them down through hydrolysis reactions, you get these individual monomers, and then you rebuild them into the polymers that you need. So give yourself good fuel and you will be healthier, you'll be happier, you'll live longer, you'll be more alert and alive than others who don't. Well, I have this here. We're going to go over a couple of these prompts with respect to the essay questions that you're going to see on your exam. We'll do that later. Just remember, and I don't want to give you a guilt trip because everyone needs to live their life and eat what they want, but anything in moderation is proper. But if you eat things like this all the time, what do you think you're going to look like? And do you think that you're going to be healthy and happy? There's definitely options that are out there that are a little bit more healthy. And the irony that I've found over the years is that these things that you see on the screen are actually cheaper than the other stuff, the more processed, more fast foody type stuff. You could have something like this, or you could have something like this. And just know that everything has a consequence. Every time you eat a salad, it has a good consequence. Every time you eat something like this, it has a negative consequence. Now, I'm not saying you can't eat this, but I'm saying if you do this breakfast, lunch, and dinner every single day, you are going to start to have some effects from it. Or if you do this breakfast, lunch, and dinner every single day, or maybe a balance between the two is probably the most healthy situation. I remember I was in graduate school for 3.5 years. I was trying to get my PhD in physiology, and I remember the two things. Number one, that I had a yoga class, which I really liked. And number two, I remember leaving the University of New Mexico thinking, if I could distill down everything that I just learned, it would be to eat more and a variety of vegetables. That was it. I mean, I studied all this biochemistry that I had 700 level courses. I had courses with 3 students or 4 students and a professor for the whole semester, all these super specialized things. And then I just realized that everything that we have to keep us healthy is right in front of us. So let me give you 3 tips if you're worried about disease in your future. Because what science has found is that mostly anyone's predisposition for getting it, anyone's probability of getting a disease really doesn't have much to do with genetics or as much to do with genetics as their choices during their life. So I'm going to give you 3 tips that you can walk away with, and you're going to forget my name. 5 years from now, someone's going to say, what'd you take in high school? And you'll be like, I don't remember a single thing. And 10 years from now, you won't even remember this room or who you sat next to, and I'm okay with all of that. But what I want you to do is I want you to go away with some things that you can bring forward. And that's fine. Forget about me. That's fine. It's part of my business. I enjoy being with students now, and I enjoy being in their lives. But I don't remember who my high school teachers are. I don't remember a thing from high school at all, except for screwing around outside of the classroom. But inside the classroom, do I remember a lecture or a thing I read in the book? No way. So it doesn't matter. Take away these tips. Here's the first one. When you go to the supermarket, shop the perimeter. I want you to think about any local market like the Avon City Market. Think about what's on the outside of the store. The perimeter of the inside of the store. You have your vegetables, you have your meats, you have your dairy, you have your all these things that are less processed. Let me just put it that way. So to fill your cart first and then go through the middle aisles. This is a recommendation that I've used for a long time. If you look at the middle aisles of any store, you're going to see all these cellophane wrapped and bright colored stuff that's super processed. And this brings me to my second tip. Less is more. I had this hippie student a few years ago, and she had a hippie mom who said to her when she was like 4 or 5, she told me this. She said, my mom said that if I can find anything in the store with 4 or less ingredients, I can put it in the cart and she'll buy it. And my point is if you start looking at labels and you can't even read what's on the label and pronounce the words, please stay away from it. It's probably not very good for you. Finally, boys, ketchup is not a vegetable. And the vegetables that you do get, like mushrooms or olives or something on the top of a pizza, that's not enough for your recommended weekly allowance. So I recommend, third tip, get a cutting board. I get it, you're going to go to the dorms, but when you get out of the dorms and you get your own place, get a cutting board, get a skillet, get some oil, and that's all you need. And then just experiment. Every time you go to the store, grab a brand new vegetable, chop it up, look it up on the internet how you're supposed to cook it, and you'll be surprised sometimes, but probably not surprised other times. But the point is get a cutting board. That's the first step here. So those are my 3 tips. Well, I've asked around for years, what is the world's healthiest food? Like if you could just choose one food item. And some people say it's Vegemite, which has like this corollary in England called Marmite. Apparently it has a very distinct taste. I've had Marmite, but I haven't had Vegemite. Have you had Vegemite? Apparently you either love it or you absolutely hate it. You should look at the face of someone who you ask, hey, do you like Vegemite? And either they'll light up like a Christmas tree or they will look like they're going to die. So the point is, try the Vegemite. My colleague David Frankel, he told me that brewer's yeast might be the world's most perfect food. I've read that quinoa is the world's most perfect food with all these essential amino acids. It's got all the complex carbos that you need. It has these good fats. And I read an article once about Pizarro and his men as they were going through the Sacred Valley outside of Lima, Peru, with gold fever in their eyes. And they were trampling all these fields to go and kill these Incan people in the Andes to try to pillage and plunder all their gold. And the point of the article was, yes, he found some gold. Yes, he brought it back to Europe. But had he just bent over and taken the seeds that were at his feet of quinoa and brought it back to Europe, Queen Isabella or whoever, I don't remember, maybe he was Spanish or under the Spanish flag, they would have made like 7 times more money over the hundreds of years after that than the gold that they got. And it would have nourished many more people. So there's one more almost perfect food, and here it is. It's going to be a movie.
Every year, malnutrition kills 5 million children. That's 1 child every 6 seconds. Despite those heartbreaking statistics, we actually found a good news story about malnutrition. As we reported last fall, the Nobel Prize-winning relief group Doctors Without Borders says it finally has something that can save millions of young lives. It's cheap, easy to make, and even easier to use. So what is this miraculous cure? It's a ready-to-eat, vitamin-rich concoction called Plumpy Nut, an unusual name for a food that may just be the most important advance ever to cure and prevent malnutrition.
Okay, so I'm not going to tape Plumpy Nut onto this video, but I would like you to watch it. Obviously, that's Anderson Cooper, a little bit younger, but I have found some recent articles suggesting that Plumpy Nut is still working its magic. I'm going to ask you about Plumpy Nut later, so please watch it now. Or, sorry, I'll ask you on a later date. Plumpy Nut, not head of a baby. So let's talk about nucleic acids here. And remember what we're talking about here are DNA and RNA. These things store and transmit hereditary information. What does that mean? That means that there's information that is inherited, that's hereditary, inherited by one generation from the previous generation. And that's where the information is stored. We remember that the information that is contained in DNA is transcribed into packets of information that we call RNA, and then that is translated, or translation occurs, then a sequence of amino acids is created, and then eventually a protein is created. And we've gone over this central dogma before, but I'm going over it with you now because I want to ensure that you've got this down. Make sure you got it down because it's going to govern a lot of what happens once we get through the energy portion in chapters 8, 9, and 10 this semester. Well, let's go here to this little summary page of all these things that exist in chapter 5, specifically right here where it says concept 5.5, nucleic acids store and transmit hereditary information. Just understand that each of these monomers that are put together to make a polymer that we call DNA or RNA have 3 components. They have a phosphate group, they have a 5-carbon sugar, you can see that in green, and then they have a nitrogenous base. And all that means is that the base has nitrogen. And these are the 4 bases. We've got them up here, A, T, C, and G. You've heard about those before. Those are just abbreviations for what humans call these chemicals. And we'll get to that in a second. So please remind yourself of the difference between DNA and RNA. Remember, DNA is usually double-stranded. DNA has a nitrogenous bases of C, G, A, and T. And then they have deoxyribose as their sugar. Conversely, RNA is usually single-stranded. U, or uracil, is substituted for thymine, or the T, in RNA. And then also it has ribose as its sugar. And I'll get to that distinction in a moment. You can see that DNA stores the hereditary information, and some people say that RNA is really kind of the worker here that takes the information from DNA, and then that's translated to become a sequence of amino acids. So make sure that you understand this flow, and now let's get to the detail. Go ahead and press pause and go over or speak out loud, please. What is happening here? What do you see in this picture? Well, basically what I see is transcription, where in the nucleus, the message on DNA is being turned into a message. It's being transcribed like someone with a transcription of events, and then someone's going to read their transcription of events, and that's the RNA. It's going to go out to the cytoplasm. See that DNA direct synthesis of mRNA, and through mRNA it controls protein synthesis. So DNA is pretty important, but I like to look at it as like a book. It's closed, and then it gets opened. Someone transcribes what they see, but then remember, DNA doesn't do anything. It then gets closed again and hidden away in the nucleus where it's nice and protected. It's the RNA, which is that working copy that then someone is going to translate. And that someone is a ribosome. You can see that on the screen, and you can see the amino acids coming in there into sequence. That's the translation. So protein synthesis occurs in ribosomes. We'll see that in a little movie later. We'll get into it in much greater detail later on this semester. Find this picture. There's a lot of good information here. This talks about the structure of nucleic acids. You can see over there on the left the polymer of a nucleic acid. That could be RNA. It looks like it's single-stranded. You've got those 3 components there in the middle box. And then we'll talk about the sugars and nitrogenous bases on the right side. Well, this is called a nucleotide here. And those things get added together. What type of reaction do you think is going to happen when they come together to make a dinucleotide or a trinucleotide or a polynucleotide? That's right, a dehydration reaction, just like all these other monomers into polymers that we've seen in chapter 5. Each nucleotide consists of a nitrogenous base. Penta means 5 carbons. You can see that there's 4 carbons there in the structure, and then 1 carbon outside off of the number 4 carbon. So if you remember, if you count here, and that's an oxygen atom in the pentoses that we see on the screen. So this is carbon 1, 2, 3, 4, and then out here is this C, this carbon. It's off of the 4' carbon. Remember, there's a phosphate group there. Also, you learned about those in chapter 4, those different functional groups. Well, let's look at the differences here between these two types of sugar. So in DNA, we have deoxyribose sugar. What do you think deoxyribose means? No oxygen or oxygen less on the ribose. And you can see that right here. Make a mental note or write yourself a quick note that it's on the number 2 carbon where the oxygen is not there. I have no idea why this is and why the sugars aren't the same between DNA and RNA, but they are just different. So I'm sure someone knows why this is, but there's no oxygen here on the number 2 carbon of RNA, so that's called deoxyribose. And then on the right-hand side here, you see a ribose sugar, and that's on the number 2 carbon. You see that it does have an oxygen. Well, let's talk about the distinction between these different nitrogenous bases. Up here at the top, you see the pyrimidines. Students have told me over the years that they memorize this a few different ways. Number one, they would say pyrimidines has a Y in it, and if you notice, cytosine and thymine both have Ys also. So that's an easy way to remember the 2 pyrimidines. Uracil is going to be an RNA, but it's just with respect to DNA, that's how they remember it. Also, some students have told me longer name, smaller structure. If you notice, it's just a single ring structure for all 3 of the pyrimidines. A single 6-membered ring. So if you go down here to the purines, down here at the bottom, I always think of it as AG, which is the abbreviation for gold in the periodic table. So AG is gold, and that might be pure. Someone would also say, well, it's the shorter name, purines versus pyrimidines, and it's the larger structure. So that's how they've memorized. 6-membered ring fused to a 5-membered ring. You see a lot of nitrogen atoms in there. That's why it's called nitrogenous bases. And these things are going to line up here across the middle of DNA anyway and create hydrogen bonds with their counterpart on the other side. I'll show you that in a few minutes. Well, if we go to this essential knowledge sheet from the College Board and we see number 1 here, go ahead and press pause and read this, and you should be able to draw me this right now and on the test day, and then write me a paragraph on it. Tell me what's going on here with all these things. Don't assume that I know what you're trying to say. Say it in science writing. Well, when we link these things together to build a polynucleotide, you're going to see that they're bound by covalent bonds. I've kind of drawn that up there. I would point to it on the board, but I guess if you look along the left side here, those circles that are filled in, those are phosphate groups, and you can see that there's just these straight lines, and that's representative of sharing of electrons through covalent bonds. So basically, one ribose sugar is bound to the next ribose sugar through this phosphate group and covalent bonds to to the phosphate group. Let's put it down. You can see that here. It's coming off the number 3 carbon of one of the riboses, and then now down below it, it's bound, so that's it's on the 3 carbon of the sugar and the phosphate group, and then it's bound to the 5' carbon on the next ribose sugar, and you can see that. I'm going to have you draw this in a couple minutes, but first, have you ever wanted to go to MIT?
Hi, in this clip we're going to discuss the numbering and labeling conventions of RNA and of protein. Here we have a nucleoside triphosphate, or NTP. This NTP is numbered on each carbon, and we're going to start here to the right of this oxygen and label 1' and proceed 2', 3', 4', and this last 5' carbon. This means that when we polymerize 2 or more of these NTPs to get an RNA chain, it becomes apparent why one end is labeled 5' and the other end is labeled 3'. On this end, the extending atom is the 5' carbon, and on this side, the other end, the extending atom is the 3' carbon attached to this oxygen. This 3' carbon is where you would attach another NTP if you were going to extend this chain. And so we label the ends of RNA 5' and 3', and RNA is always polymerized in 5' to 3' direct.
That's super important to know. Thank you, Sarah, for that. You ever seen a chalkboard before? I remember chalkboards when I was growing up, and then I remember my parents saying that when people would get in trouble, they would have to clap the erasers to get rid of all the chalk that was in them. There's some math teachers here that said that they still use chalkboards down at CU, especially in the math lectures, and that their professors by the end of the day would just be covered in chalk. Awesome. What do you notice about the directionality of each strand of these polymers? So let's imagine, so we've got this DNA, and it's going to then eventually be, the information from it's going to be used to create an amino acid sequence. But you can see here that one side of the DNA is going from a 5' to 3', meaning if we were going to add another nucleotide, we would add it to the 3' end. If you look at the other side of the DNA, you can see, I wish I could point at things here, you can see that it's going in the opposite direction. Isn't that weird? And I've kind of drawn it up here as these things being upside down. Do you see that? So I'm going to write up here on the left side that this is the 5' carbon, this is the 3' carbon, and so what would that be on the other side of this DNA helix? Well, this would be the 3' carbon, and this would be the 5' carbon, and the idea is that it would actually be read this direction. This one would be read in this direction. That's called anti-parallelism. They're parallel, but they're anti-parallel. I think it's crazy how DNA grows, and this is going to become very important when in chapter 12, when we talk about DNA replication and cell replication. You got to make new DNA every time you make a new cell, and they just grow in opposite directions. It's really weird. But they have a buddy that they always consistently bind up to. A always binds to T, and C always binds to G. In the DNA double helix, the two backbones run in opposite 5' to 3' directions from each other, an arrangement that we call anti-parallel. I'd like you to pause. It's that important right here. Spend 5 minutes and draw this thing. Please focus on the exterior, the outside of the ladder, or the sides of the ladder that just repeat themselves. It's a ribose sugar phosphate, ribose sugar phosphate, ribose sugar phosphate. That's all you see here. And then the rungs or the middle of the ladder, you'll see that the bases are going across there. So I'll be quiet. Please draw. Well, something that I want to point out to you is that between the two nitrogenous nucleotides that we call adenine and thymine, you see that there's 2 hydrogen bonds there, but you also can see that there's 3 hydrogen bonds between cytosine and guanine. These are more strongly bound to one another through these hydrogen bonds than the A's and the T's are. Now, any hydrogen bond is super weak, and we have learned that in the whole water thing, that they're constantly breaking and reforming. So individually weak, but collectively strong. If you have a chromosome, like chromosome 1 has like 250 million letters on each side. If you've got 250 million of these pairings of nitrogenous spaces, and then each of them have all these hydrogen bonds, collectively very strong. Well, make sure you're able to talk about this in an essay, draw this. I would say just reproduce this thing, give me a paragraph, tell me what you know about 5' to 3' and so on.
In DNA and RNA molecules, nucleotides are strung together in long chains formed by covalent bonds between the sugar of one nucleotide and the phosphate of the next. RNA usually occurs as a single strand formed in this manner. DNA, on the other hand, is usually found in the nucleus of cells with 2 strands bound together by hydrogen bonds between the nitrogen bases. However, only adenine and thymine bases and guanine and cytosine bases can bond together. Because of the different chemical bonds and monomers that make up DNA molecules, they take on the appearance of a twisted ladder, or what scientists call a double helix. DNA molecules can be millions of nucleotides long. Each long, complex DNA molecule in the nucleus of a eukaryotic cell is called a chromosome. Chromosomes contain subdivisions called genes. The sequence of nucleotides in most genes contains a kind of biological Morse code that has the instructions for building one complete protein. As we saw earlier, these instructions are copied onto RNA molecules that travel into the cell's cytoplasm and there direct the synthesis of proteins by ribosomes.
Okay, so the final concept here in the chapter has to do with comparing DNA sequences. You can imagine that if you took a cell from a chimpanzee and a cell from a human and you looked at the DNA, that it would be that the DNA sequences would be similar. If you took 100 random letters from the human DNA and 100 random letters from chimp DNA, I'm going to tell you that some will argue that it's 97 to 98% identical. If you were then to take a cell from like from like a snail or a bacteria, do you think that the DNA would be a little bit different than the DNA of the chimp and the human? And my answer would be yes, because DNA is what dictates amino acid sequences, which dictates proteins, which dictates the form and function of organisms. And so the point is that DNA mutates through time at a very regular rate, and at the end of this, got it right here. At the end of this chapter, in the 10th edition, pages 88 and 89 talks about using DNA as a tape measure for evolution. So the idea is that we could say, well, these things have 97% commonality between their DNA sequences. But now this and this thing have 41% commonality between their DNA sequences. And then we could go back in time because we know how long generally, on average, it takes for DNA to mutate with a stable mutation, and how long it takes for things to evolve, that we could say, well, then a human and a snail, because they only have 41% commonality in their DNA, they must have had a common ancestor and diverged from one another in the evolutionary tree of life, history on this planet, X amount of million years ago, let's say 40 million years ago, or whatever it is. And we can do that if we compare DNA sequences. And that's what you're going to do here in 5.6. And I want you to spend some time on this. I'm going to ask you about this on the exam. This is me pushing you a little bit, not spoon feeding you everything. So make sure that you read this, ponder it, and we will talk about it. I will talk about it in class if we have class. But the point is you will see it on the exam, and I will debrief with you, I promise. What I would ordinarily do, and maybe I'll do this if I see people in person, is I would have kids take 4 blank pieces of paper and just write down everything they know off the top of their head about each of these 4 major macromolecules. And then I would have you share with your neighbor and discuss. And then basically this will serve as one of 2 things. Number one, something that gives you confidence. You'll say, wow, I really know a lot about carbohydrates off the top of my head. Or you'll say, okay, this is the one I'm weak on, or weak on 2 out of these 4. And then really it'll show you what you don't know, essentially. And you really need to start to buckle down a little bit more. This test is not easy. It's as big as the first one, and it's going to be hard. Good luck. Let me know how I can help you. Make sure that you don't sleep on the Cliffs Notes. The Cliffs Notes assignment is very big for this chapter 5, and there's going to be vocabulary words as well. Well, do not write answers to these, but you can certainly take a look at them. They're the learning targets that I would have had you do in class. Okay, thanks, everybody.